Hi, biodiversity monitoring is essential for conservation efforts. The success or the failure of any conservation practice is measured by monitoring trends in biodiversity. So in this session, I am going to talk more about how biodiversity is being monitored. So there are different criteria for the effective monitoring of biodiversity. The central point is that when we are monitoring biodiversity in a small geographical area, it's very easy because we can have an in-depth study of changes in biodiversity. We can study almost all parameters if it is a small geographical area. But if you are studying a large area or in biodiversity conservation efforts, we basically interested in global biodiversity changes. It will become very much difficult. We cannot have an in-depth study in all over the world. So it will become so much more complicated. In this scenario, we need to have some consistent methods which can be used repeatedly for monitoring diverse trends in biodiversity. The methods we are using for one area cannot be used in another areas. The methods used for monitoring biodiversity in tropical rainforest is way much different than the methods used for studying marine environments. Or maybe deserts need to have a different methods. But at the end of the day, we need to have some mechanism to unify all these different types of measures in a single platform so that we can measure or we can represent the global biodiversity, or we can say the global biodiversity trends, whether it is increasing, whether it is decreasing, or our conservation practices are effective or not effective. So in order to have such decisions or such indication, we should have a unified way of biodiversity measurement. For that, there are different criteria need to be satisfied when we are going for a biodiversity monitoring. Here I am pointing out 5 different criteria which is essential for biodiversity monitoring. First one, representative sampling locations. Especially in case of a global biodiversity scenario, we should have representative sampling locations. Sampling locations should be random and it should be equally distributed all over the world. So the selection of sampling locations is very much critical. So either we can go for a design based sampling locations or model based sampling locations. So if we have a forest for sampling, we can go for randomized sampling. So first thing is sampling should be representative. For that, the sampling should be randomized. Secondly, sufficient sample size. The sampling size, the number of samples we are taking from a particular geographical area, should be enough to represent the biodiversity. If the number of samples we are taking in a location is less, the precision will be less, the bias will be more. We will get unreproducible data. The data cannot be reproduced because of the errors in that. So we should have representative sampling from which is spread across the geographical area, and it should have sufficient sample size so that the data is not biased. Thirdly, sufficient detection of target species. Each and every species have a detectability. Some of the species are disease can be some of the organisms can be detected easily. If you are looking for the plants, they won't move, so it is pretty much there all the time. You can actually like uh, document it at any time. They are not mobile. But sometimes other type of animal, in case of animals, they are more mobile. They will have maybe have a seasonal variations, or maybe some of them are active in daytime, others may be active in night time. So there are so many deviations are there depending on the type of species. So what I am telling is like each and every species have a detectability. Some of them are easily detectable, some of them are not easily detectable. In case of animals, if you want to document the population of elephants, it is very much easier than documenting the presence of an insect. So the detectability for the elephants is high, but detectability for an insect, it's very less. So in order to have a good biodiversity measurement monitoring, we should consider the detectability of the species. Okay, then sufficient detection of target is essential. Even if we have enough number of samples and we are doing in a randomized way, the detectability of species is also important. If the organism or the biodiversity species we are looking for is not that much detectable, we should have devised some methods to overcome the limitations. Fourthly, representative samples of all species. Here we are talking about the global biodiversity trends. The biodiversity include different species at different taxa levels. So in order to have a holistic view of the point, we should have representative from each taxa, representative groups from all the groups. We should have the groups from the plants, maybe from the amphibians, from birds, different types of bacteria and microorganisms. So in order to have a precise measure of global biodiversity trends, the monitoring system have representatives from all the species. Okay, so that we'll get a better idea about the global biodiversity trends. Finally, a temporal sampling scheme designed to aid valid inferences, because the most of the organisms has temporal variations. They have seasonal variations, and they may be active only in some types of some time of year, some seasons. So our sampling methods should be sufficiently long so that it will include all the species. So these are the five main criteria for effective monitoring. They include representative sampling locations, sufficient sample size, sufficient detection of target species, representative samples of species, and temporal sampling schemes. So this is how the whole biodiversity monitoring thing work. Actually, this has three different components. First one is the data collection. Secondly, we have EBVs, and finally we will end up with indications and strategic plans. So in the first stage, what we are doing, we are using different techniques to actually document the biodiversity. Either we can use molecular biology techniques, there are satellite based techniques, maybe we can go for an expedition, especially in the sea. Then there are advanced techniques like high resolution imaging, or maybe we can use observations, bird watching, things like that. So by using all these techniques, we are actually generating the primary data about the biodiversity. So the first stage is generating data, getting the primary data from the field. Okay, using different techniques, we will get different data about the abundance, about the diversity, ecosystem function. So first stage is generating primary data. Okay, it's a field study. Secondly, we go for EBVs. EBV stands for environmental biodiversity variables. So whatever data we are getting, we are converting it into EBVs. EBVs include essential biodiversity variables, include the genetic composition that represents the allelic diversity, species population traits, the species diversity, then community composition, ecosystem structure, ecosystem function, and ecosystem services. So the thing is that in the primary data collection, we are using different techniques. In the different conditions, different areas need different techniques. We cannot use the techniques what we are using in the marine environment in a tropical rainforest. So the techniques will change, the methods will change. But whatever techniques we are using, we are converting this data into EBVs, essential biodiversity variables. So we will get data about the allelic diversity, species diversity, community diversity, ecosystem structure, function, and services. So the EBV is actually a intermediate in this flow. First we will get the primary data, we will convert the primary data into EBVs, and EBVs actually represent the biodiversity. Diversity at allelic level, species level, community level, ecosystem level, and ecosystem services level. So these EBVs are generated in association with the biodiversity networks. So there is a constant knowledge exchange between the EBVs and the biodiversity observation networks. The biodiversity observation networks are called bonds. There are thematic bonds. Thematic bonds means they are based on particular themes like bird watching or butterfly watching. Groups are there, then national and regional bonds are there, the national level biodiversity observation networks. They will exchange data constantly with the to generate EBVs, essential biodiversity variables. So from different sources, from primary data collection and biodiversity observation networks, we will get idea about the biodiversity, essential biodiversity variables. And each and every year, how the biodiversity variables are changing is important. Based on the changes in essential biodiversity variables, we will calculate indicators of biodiversity. So these indicators indicate the changes in trends in biodiversity, whether the biodiversity is increasing or what way it is increasing, or how the conservation efforts are affecting the performance of EBVs, or how the climate change is affecting the EBVs, which is the measure of biodiversity. So from these EBVs, we will calculate, or we will find the indicators of biodiversity changes, and these indicators are used for strategic plans, for the conservation efforts, policy making, law making. Everything is based on the indicators. These indicators indicating the changes in biodiversity. So simply say, we have first step in biodiversity monitoring is primary data collection. From that we will derive essential biodiversity variables. The performance of essential biodiversity variables is used to calculate indicators of biodiversity, and strategic plans, law, and everything is made based on the changes in these biodiversity indicators. So the middle of this flow is EBVs. EBVs stands for essential biodiversity variables. It is used to optimize biodiversity monitoring initiative. It is actually formulated by a group on Earth Observations Biodiversity Observation Network, GeoBond it's called. It is the basis of efficient and coordinated monitoring program worldwide. It's actually helped to integrate different monitoring programs which is happening all over the world, because any type of biodiversity monitoring you are doing, you can actually convert the data into essential biodiversity variables. So if you are observing the changes in the elephant population, we can actually convert this data into one of the biodiversity monitoring biodiversity variable, like the abundance data, the population dynamic data. So we can convert any type of study into a single platform. Okay, that's what the EBV stands for. Okay, it identifies minimum set of variables that indicate global biodiversity changes. So it's actually a very good platform for to measure the global biodiversity changes. Whatever type of study you are conducting, you can actually convert your data into one of the essential biodiversity variables. Based on that, we can actually calculate the performance of biodiversity, or the trends in biodiversity. So this is how it look like. What we need for the global biodiversity measure monitoring is the indicators of biodiversity. Okay, we should have some mechanism to say that the biodiversity is increasing, decreasing, or whatsoever. So what we need is indicators for biodiversity changes, and what we have is this type of different types of studies. So EBV is actually in the middle of these two, our primary data and the indicators. So it actually helps to integrate the primary data into indicators. Here we can see the arrows are going everywhere, and we will get some type of other type of indicators. So same like the previous image, we have primary data, EBVs, and indicators, and based on indicators we are making policies and laws and etc. So the primary data can be classified into 4 different types: extensive scheme data, intensive scheme data, ecological studies, remote sensing. We already talked about extensive and intensive studies in biodiversity inventory. Extensive studies are used for huge, large geographical area. It is not that much detailed study. Intensive studies, on the other hand, it is done only in selected areas. It is a comprehensive study of all biodiversity parameters. And then we have ecological studies. Ecological studies can be a field study, or it's an experimental study, like the ecotron experiment. So ecological studies are also a type of primary data collection. Then we have remote sensing. which is also included in primary data collection method. So we have 4 different types of primary data collection methods. From this, we can derive EBVs, essential biodiversity variables, in the form of allelic diversity, species diversity, ecosystem structure and function, and primary productivity. Primary productivity is basically an ecosystem service. So we can actually, genetic composition is allelic diversity, species diversity, ecosystem structure, and ecosystem function. These are the EBVs and EBV classes. So if you are studying an ecological study, it can get data for all the 4 different types of EBVs. Okay, you can see the difference in the thickness of the arrow. Ecological studies mainly contributed to the species population or species diversity. Maybe the remote sensing can give data about the ecosystem structure and ecosystem function. Intensive schemes can produce data to derive all type of EBVs. Extensive studies are basically about the species abundance. Okay, so we are, for each of the biodiversity variables, we can pull out data from different types of studies. So in the globe, different peoples are doing different types of studies. Actually, by using this scheme, we can integrate all of this data into a single platform called essential biodiversity variables. And we can actually calculate the performance of each of this thing, how allelic diversity is increasing or decreasing, species changes in species diversity, etc. So from this, we are deriving indicators. Indicators like LPI, RLI, extent of forest and forest type, etc. So some of this, if you know the ecosystem structure, we can derive these things. If you know species diversity, LPA and RLI can be derived from that. So whatever type of primary study you are studying, you are conducting, we can integrate it into EBVs, and based on EBV we can calculate the indicators. Indicators are maybe numbers or maybe plots which represent in the trends in biodiversity. So EBV stands between primary observation and high level indicators, like this. This is the primary observations, these are the indicators. In between we have EBVs. Only using EBVs we can actually integrate the primary observation into indicators. It represents essential aspects of biodiversity or integrated it to deliver high level indicators. It actually covers the data gaps and optimize the use of available data. There are different classes of EBV. The EBVs can represent the genetic composition, species population, species traits, community composition, ecosystem structure, and ecosystem function. And we have, of course, we have different schemes to integrate the primary data into different types of EBVs. And talking about the primary data can be validated or characterized by different types of limits. Like some of the primary data are more useful to generate EBVs than the other. The primary data, like the type of primary study, the primary data study can be done by extensive schemes, intensive schemes, or remote sensing, or field experiment, things like that. And each of this class of experiment have different types of coverage. Here we have 3 different types of coverages: geographic coverage, taxonomy coverage, temporal coverage. Okay, and different type of study will have different types of coverage. Maybe one type of study may have the higher geographical coverage, maybe the temporal coverage will be less. Maybe taxonomy coverage is high and temporal coverage is less. Things like that can happen. So we should understand what is this different types of coverages are meaning. First one is geographical coverage. Geographical coverage is how much area you actually covering. If you are looking for the tiger population, how much area you are covering. So if you want to study the population or the population density of the tigers in India, so if you are conducting study only in a limited area, your data may not be that precise. But if you are doing a study which cover all the possible locations for tiger inhabitation, it will be a better study. So geographical coverage is actually meaning 2 different things: how much area it is covering, how much samples you are taking. If you are covering a huge area but you are taking only few samples, you may end up in false data. So for a geographical coverage, there must be a good coverage of the area, as well as the sampling density should be high. You should get more and more samples. As you take more and more samples, the precision of data and the chance for bias will be less. So geographical coverage need higher coverage, higher area-wise coverage, as well as the sampling density. You should have more and more samples to get better data. Secondly, the taxonomy coverage. Whenever we are monitoring biodiversity, we are not monitoring for a single species. We may be looking for different species at the same time, so that we will get an idea about the trends. So better coverage of different representative from all taxonomic groups, we should have more sampling frequencies. So okay, the sampling frequency, number of samples you are taking should be high. Maybe you can take the samples in a day, or maybe you can take samples one day in every week, maybe one day in every month. Things like that can be done. So if you are taking more and more samples, the sampling frequency will be high, and more and more taxonomic groups will be included in your data, so that it will be more representative data. And finally, we have the temporal coverage. Temporal coverage means how much the length of time the study is carried out. If you are doing your study for only for 1 hour, the chance of documenting all the available species in that area is very less. Some of the animals are nocturnal. You can only get the data about that animal if you are conducting the study at night. Maybe some of the insects have seasonal variations. They appear only in few months of the year. So if you are not conducting your study in that particular month, you will get no data. So for a perfect study, there should be geographical coverage, taxonomy coverage, as well as temporal coverage. So these are the three major criteria to assess the performance of a primary data collection. And two other terms related to this is sampling efforts. The number of ecosystem variables or functional groups are monitored. If you are measuring only less number of less number of species or the functional groups, your data may be more biased. If you are taking into consideration different groups of animals, different groups of ecosystem function, your data will become more and more representative. So sampling effort is another factor. If you are having more and more sampling efforts, the data will be better. And finally, we have time series, just like the temporal coverage. The time of study, the duration of the study should be maximum. But when we are trying to integrate the geographical coverage, taxonomy coverage, and temporal coverage into a study, there will be factor of cost, factor of labor, and factor of efforts needed for the processing these all data. So this is even though we need to have the maximum coverage in geographical, taxonomic, and temporal level, but there are so many factors which limiting the coverage of any primary data source. Thank you so much.